Thank you. Okay, so as Mike told, I am going to present some of my previous research that I performed during my PhD. The title of my PhD dissertation is Response Capacity to Environmental Stress on Pinus sylvestri and Pinus nigra populations at the southern distribution limit, a multidisciplinary approach. So first of all, I will give you a little introduction about the context of my study, the study area, target species, objectives, and working hypotheses. Climatic change is one of the most important concerns for ecological science nowadays, and through its effects on species physiology, phenology, and distribution, could change ecosystem structure and function. In this context, species-specific differences in vulnerability to environmental stresses could change the composition and structure of the vegetation. Thus, in a hypothetical community formed by 2-3 species, if one of the species are more drought tolerant than the other, this could promote a change in dominance towards the less vulnerable species under a scenario of recurrent drought. However, it is very important to take in account biotic interactions such as herbivory, because biotic interactions can modulate the species responses to climate. Thus, if the less vulnerable species is strongly selected and damaged by herbivories, climate-induced change in composition can be slowed down or completely inhibited. However, if the more vulnerable species to drought is the species selected and damaged by herbivories, the climate-induced change in composition could be boosted or speeded up. Many of these changes of composition could occur at the southern distribution limit of a species, when occur rare age populations. These populations occur far from the optimal environmental conditions of a species and also occur close to the species tolerance limits. Thus, the study of the ecological dynamics of species borders could provide new insights in the potential ability of a species to respond to climatic change. Under the scenario of climatic change, it is expected that the extreme climatic events such as extreme droughts or heat waves increase their frequency and intensity. In the last year, some studies recorded some vegetation change in response to these extreme climatic events, but this is not always the rule. Sometimes there are some stabilizing mechanisms that can reduce the impact, the negative impact of the extreme events, or boost the recovery of vegetation after these disturbances. So here we have some of them, some of these stabilizing processes. Tolerance to stress can be due to the local adaptation or a variety of existing genotypes that respond in different ways to changes in climate. Then we have phenotypic plasticity that allow individuals to modify the form and function in response to changes in environmental conditions, particularly changes in climate. A small-scale variation in environmental conditions could provide some hotspot of survival or regeneration for a species after a disturbance. Facilitative interactions such as nurses wraps can boost regeneration also after a disturbance or during dry periods. And some of the new environmental conditions that arise because of the climatic change or that arise after the extreme events could be beneficial for vegetation. For example, after a massive tree mortality event, there is a less tree cover. That means less competition for resources for the remaining of trees. So this could be beneficial for these remaining trees that are alive. Also, increasing temperatures could be beneficial. For example, if the spring temperatures increase, they can be the beginning of the growing season can be advanced, and buffer at least in part the negative impact of increasing temperatures in dry seasons. Okay, it is important also to take in account the importance of the different ontogenetic states, because if we want to analyze the response of the population to an environmental change, the response of the population will be determined by the integration of the response of each ontogenetic states. In my researches, I focus on saplings and on adults. Saplings are not well described in Mediterranean environments, and are very important because established pre-reproductive saplings represent the near future of the forest. So my study area is located in south of Spain, in the Granada province, and I sample in 2 locations. One in Sierra de Baza, is a natural park, and here I study the radial growth climate relations in adult pines, and also vegetation responses to extreme events. And in the other location, in Sierra Nevada National Park, I study drought-related mortality in pine saplings. So Sierra Nevada, it is a national park, represents the absolute southernmost distribution limit for Pinus sylvestris, and hosts relic population of Pinus sylvestris subspecies nevadensis, that is located around 1,800 meters and 2,100 meters above sea level. There are also widespread reforestation composed by Pinus sylvestris and Pinus nigra, and the annual precipitation is around 200 and 900 millimeters. In this area, we have very marked dry season in summer for June, July, and August months. And the other study area is Sierra de Baza Natural Park, also in Granada, hosts some relic population of Pinus sylvestris and Pinus nigra, located at 1,500 and 2,100 meters above sea level. And this is a drier area than Sierra Nevada. The annual precipitation is 482 millimeters, and we have also a very marked dry season during summer. Okay, the target species are Pinus sylvestris and Pinus nigra. Pinus sylvestris is a boreal species distributed mainly in North and Central Europe, and usually occur at colder and more humid locations than Pinus nigra. Pinus nigra is a Mediterranean species distributed mainly in south of Europe, and often occur in drier and sunnier locations than Pinus sylvestris. Sierra de Baza and Sierra Nevada populations represent the southernmost distribution for this species, although we have some population of Pinus nigra in North Africa. This is a nice picture of Pinus sylvestris forest in Sierra de Baza. This is a species that have 2 needles per fascicle. The needles are quite short, the cones are quite small, and have the bark of the upper part of the trunk and the upper branches are orange color. This is very good characteristic to identify this species. And then we have some Pinus nigra saplings and some adults. The bark is silver gray, have 2 needles also per fascicle, and the needles are longer than in the case of Pinus sylvestris, and have also bigger cones. Okay, the main objective of my study is to analyze the response capacity to environmental stress, mainly climate and herbivory, of Pinus sylvestris and Pinus nigra populations at the southernmost distribution limit. However, in this talk I will deal mainly with climate responses. And we work with 2 alternative hypotheses. So in my studies, I analyze these populations located at the southernmost distribution limit. So these populations are close to the species tolerance limit. So the 2 alternative hypotheses is, one is this population will be very highly vulnerable to climatic change, or stabilizing processes will buffer the negative impact of climatic change. So we work with these 2 alternative hypotheses. The first work that I am going to present deals with a pine sapling mortality event in Sierra Nevada. And in the last year, many works report massive tree mortality events around the world, usually related with drought events or heat waves, and also involving, in many cases, biotic agents. However, the major part of this work focus on adult trees, and this work is interesting because it deals with saplings. So to analyze the response of pine saplings to the extreme drought, we use foliar carbon-16 and foliar oxygen-18 data, stable isotope data. And to analyze this data, we are going to use the conceptual model done by Grams and others that allow us to distinguish between the effects of stomatal conductance limitation and the effects of photosynthetic rate limitation. Okay, the main objective of this work is to compare mortality, growth, and physiology of Pinus sylvestris and Pinus nigra saplings in response to an extreme drought event. Also, we want to analyze any differences in the response to drought between death and live saplings in order to elucidate the mechanism behind the drought-related mortality. In 2005, we have an extreme and very severe drought in the area. The most severe drought in the last 50 years, characterized by very high summer temperatures. This drought affects a reforestation experiment, a previous reforestation experiment of a mixture of Pinus sylvestris and Pinus nigra saplings growing in the same place, and all the saplings have 10 years old. So this is a very good opportunity to address pine sapling response to a drought because we control age and environmental conditions. In this picture, we can see easily some live saplings and some dead ones. We calculate a drought index based on precipitation and potential evapotranspiration for the whole lifespan of saplings, and we can see that the 2005 conditions are very dry. We also calculate mortality rate for both species, relative height growth. We calculate relative height growth with measuring the height of the sapling in different times. For this, we measure annual height growth following yearly gulls and bath scar. This is very easy in this species because they have only one flush per year. And we also collect needles for the last 4 cohorts from 2002 to 2005 to analyze foliar carbon-16, oxygen-18, and nitrogen content. So the correlations between foliar carbon-13 and relative height growth indicate that the major limitation to photosynthesis is stomatal conductance. Also, the correlations between carbon-13 and oxygen-18 points to the same conclusion, that the stomatal conductance is a very important limitation to the photosynthesis. And now I am going to show you some data of carbon-16 and oxygen-18 in dead saplings. The signal of stable isotopes in dead saplings are very informative because they tell us information about the state of physiology of saplings at the time of death. So this is really useful. We can see that in 2005, the dry year, we have a very important increment of carbon-13 in both species, and also a very important increment in oxygen-18 for both species. However, the increment in oxygen-18 is more higher for Pinus nigra than for Pinus sylvestris. If we return to the model of Grams and others, we have this situation, C, because the values for both the species, in the case of carbon-16, it's very similar, but the values of oxygen-18 are higher for Pinus nigra. This means a higher reduction for Pinus nigra for both stomatal conductance and photosynthetic rate. But we also measure nitrogen content in both species. The values are very similar, so this suggests that the photosynthetic rate is quite similar for both species. Thus, we can conclude that the stomatal conductance that undergone Pinus nigra is higher. I mean, it presents greater stomatal reduction that Pinus sylvestris. At this stronger reduction in stomatal conductance is reflected in the growth, because if we see here, there is a reduction in growth in the dry year respect to the previous year for Pinus nigra, but not for Pinus sylvestris. Respect to the mortality rate, in 2005, drought mortality rate was higher for Pinus sylvestris than for Pinus nigra, but these differences are not statistically significant. However, they contribute to a higher accumulated mortality due to drought that is significantly higher for Pinus sylvestris than for Pinus nigra. Thus, maybe the greater stomatal control of Pinus nigra could be related to its lower vulnerability to drought mortality. As conclusion, we can say that stronger stomatal control of Pinus nigra in response to dry conditions could favor a lower vulnerability to drought, and under acclimatic change scenario, lower vulnerability of Pinus nigra to water stress could promote an increment of its dominance in Mediterranean mountain pine woods. It's just a speculation. The next work deals with radial growth and climate relationships in adult trees. So, as I told previously, analyzing the ecological dynamic of species borders could provide new insights into the potential ability of a species to respond to climatic change. In fact, the rare age populations can harbor or can host critical genetic diversity for species conservation, management, and future evolutionary potential. So it is very important to address the performance of these populations. And in this context, dendrochronology could be a very useful tool. So the main objective of this work is to analyze climate tree growth relationships and tree growth variability in rare age populations of Pinus sylvestris and Pinus nigra, and compare climatic growth relationships between different environmental conditions, mainly elevation and aspect. We also want to determine changes in tree growth variability over time. Okay, this is our sampling design. We perform a sampling for Pinus nigra along an altitudinal gradient at low elevation at 1,500 meters, at medium elevation at 1,700 meters, and at high elevation at 2,000 meters. For Pinus sylvestris, we sample location with north slope and south slope. And it is interesting to know that here the high elevation population of Pinus nigra and the south slope population of Pinus sylvestris are spatially coincident, so it allows us to compare two species in the same location. You can see that the tree density is lower at the higher part and higher at the lower elevations. We sample between 11 and 17 trees in each population, and we build an independent tree chronology for each of the populations. The common period for this chronology are 1935 to 2007. We did some linear regressions between monthly climate variables and years just to analyze possible trends in climatic variables. We also, here we have the months. We also check for trends for annual total precipitation and annual mean temperature, and we recorded an increase in annual mean temperature and a reduction in annual total precipitations. For monthly values, the more strongest trends are recorded for June and July months. So there is an increasing aridity trend in the area. We have also recorded some microclimatic data along the elevation gradient for the 3 elevations. We measure minimum relative humidity and mean temperature for 2 years. The data in this slide is only for summer, I mean months of June, July, and August, and we can see that there is a trend of higher humidity at the high elevation and lower temperatures at the high elevation. And now I will go to show you some climatic growth relationship data. There is data for correlation analysis and data for response function analysis for 2 populations of Pinus nigra. Here we have months of the previous year for June to December, and from current year from January to October. So the most surprising result of this study is that we have found very little strong signals of precipitations in these trees. With strong signals, I mean when one monthly climate variable is significant for both the correlation and the response function, and we only find one strong signal in precipitation at low elevation for Pinus nigra. It's the case of May, precipitation of current May. So this is surprising because this is the dry distribution edge for this species, and we expect more precipitation signals in the other populations, but we only found this one. In the case of temperature, we find a very strong signal of previous September temperature, a negative impact of this temperature in tree growth for all the populations. Here I saw only these 2 populations, but we record it in the other ones. And we record also some positive impact of winter and spring temperatures as previous December and current February. So maybe the performance of these populations will be a balance between the negative impact of previous autumn temperatures and the positive impact of winter and spring temperatures. Finally, I have here some data of tree growth variability, specifically mean annual sensitivity. And we have here some intervals of years, and the aridity is increasing when come to the more recent data. And we can look that we have an increasing tree growth variability coupled to increasing aridity, but just in the low elevation population of Pinus nigra, and not for Pinus sylvestris or for high elevation of Pinus nigra. So maybe the lower elevation populations could be more vulnerable to future climatic changes. As conclusion, radial growth in Pinus sylvestris and Pinus nigra population at the drought-linked distribution limit is more determined by temperature than by precipitation, although for sure temperature can drive water availability. The lack of strong precipitation signal could be related to different factors. One possibility is microclimatic conditions, because we see that at high elevation the humidity is higher and the temperature is lower during summer. Local adaptation, there are some studies performed with different varieties of Pinus sylvestris, and the varieties for south of Spain invest more in roots than other varieties for northern Europe. And then forest structure properties, we see before that high elevation populations have less dense tree cover. That could means less competition for water resources. So the widespread negative effect of previous September temperature might be a result of prolonged drought stress, because September is usually the month when summer dry season ends. And the balance between positive and negative effects of temperature in radial growth could in part determine the future persistence of these rear edge populations. and the populations located at the lower elevation will be more vulnerable to future climatic changes. Okay. This is the last of my works that I will go to present, and deals with pine juniper woodlands responses to extreme droughts. Are Mediterranean pine juniper woodlands that are quite different from the pine juniper woodlands from Western United States. So in this work, we are going to analyze the resilience capacity of pine juniper woodland. Resilience is defined as the capacity of an ecosystem, community, or individual to restore a structure and function previous to a disturbance. That could be a extreme climatic event or wildfire or whatever. So to study the resilience capacity of the vegetation to extreme climatic events could be very important to forecast future changes in vegetation. So the objective of our work is to analyze the response capacity of pine juniper woodlands. For that, we focus on mine tree and mine shrub species at two different altitudinal belts. At the Euro-Mediterranean altitudinal belt, located at 1,800 and 2,000 meters above sea level, the mine tree species is Pinus sylvestris, and the mine shrub species is Juniperus communis. At the Supra-Mediterranean belt, located between 1,400 meters and 1,700 meters, the dominant tree species is Pinus nigra, and the dominant shrub species Juniperus oxycedrus. We also account here for adults and saplings to assess correctly the population response to the extreme drought event. Thus, we compare tree species versus shrub species, adults versus saplings, and also Pinus sylvestris versus Pinus nigra, where both species coexist. There are some photographs for these Mediterranean pine juniper woodlands. At high elevation, it's mainly Pinus sylvestris and Juniperus communis, but also it's possible to have some Pinus nigra. Here we have some of them. You can see the patches of Juniperus communis that at this elevation has a prostrate growth form. And then you can see other Pinus sylvestris. You can see there the orange bark, and there are other junipers and pines. And this is pine juniper woodlands at low elevations. We have here some Pinus nigra, that is the dominant species, but we can also have some Quercus ilex. This is the Juniperus oxycedrus that don't present this prostrate growth form. And some juniper and pinus. Okay, the main variable that we measure for this work is shoot growth. We measure this following yearly gulls or bath scar or differences in color and diameter. And we quantify resilience components following the methodology of Lloret and others. The drought values will be recorded in 2005, post-drought 2007 and 2008, and pre-drought values in 2003 and 2004. So resistance. Resistant is the inverse of the performance reductions undergone during the extreme drought, during the disturbance, and it is calculated as the ratio between the performance or the growth during drought and the performance during before the drought. Recovery is the ability to recover growth relative to the performance reduction, and it's calculated as the ratio of growth after drought and growth during drought. Resilience is the capacity to restore pre-disturbance growth levels or performance levels, and it is calculated as the ratio of growth after drought and before drought. And finally, we have relative resilience, that is basically resilience weighted with the performance reduction suffered during the disturbance. The extreme drought of 2005 that also occurs in Sierra de Baza was the most severe drought since 1947. So we have here comparisons of resilience components for Pinus sylvestris and Juniperus communis at high elevation, Pinus nigra and Juniperus oxycedrus at low elevation, for adults and for saplings. And in the case of resistance, we have recorded that the Pinus, adults of Pinus present higher resistance than adults of junipers. However, the junipers overall present higher recovery than Pinus species. Regarding resilience, we have here very important results because resilience values were overall quite higher because are close to 1, and 1 means total recovery, total restore of the performance levels. However, there are some differences between species, and we can see here that the Juniperus oxycedrus saplings are the most resilient ones, and significantly more resilient than Pinus nigra saplings. Regarding relative resilience, overall the junipers present higher relative resilience. That means that junipers can recover for higher reductions than Pinus species. So here we have results of Pinus sylvestris and Pinus nigra at high elevation with south aspect, where both species coexist. And we can see that the resilience is also high, because are close to 1, but there are also some differences. At sapling state, we can see that Pinus nigra are more resilient than Pinus sylvestris, and also for the relative resilience, although it's not significantly different. So as conclusions, we record high short growth resilience, close to 1, that could indicate high tolerance to extreme drought events in Mediterranean pine juniper woodlands. However, we see some differences in some of the resilience components. Adults of Pinus showed higher shoot growth resistance to drought than adults of juniper. However, shrub species display a higher relative resilience than tree species. Overall, saplings showed higher shoot growth recovery than adults. And despite this high tolerance, we observed some differences in resilience between dominant species that may alter population dynamics under a scenario of recurrent droughts. Mostly in saplings, we can see that Pinus nigra saplings were more resilient than Pinus sylvestris 1, and that Juniperus oxycedrus saplings were more resilient than Pinus nigra 1 at low elevation. And now, just to finish, I will present some of my intentions for a future research here at NIU. And the project is entitled Gulf Pinus ponderosa distribution range change in response to climatic change, demographic responses along climatic gradients. So in the last year, there are several works that record some species distributional shift in response to climatic change. And due to the predictions of the climatic change, we can expect more northward migrations along latitudinal gradients and some upward migrations in altitudinal gradients. However, there are very few studies that account for the whole species distribution range. I think that it's possible to use demographic rates as signals to detect some potential migration or some potential distributional shift. At seedling state, it is possible to measure seedling density easily to estimate regeneration capacity or regeneration potential. At sapling states, we can measure head growth, especially if the species present only one flush per year. And in adults, we can measure radial growth to estimate adult growth. It is important to account for all of these demographic rates at the same time, to use demographic rates as signals, to assess properly environmental population response. So I think that these demographic rates could be used as signals. For example, if we have one population at the species border, and we measure these demographic rates, and we measure low regeneration and low growth for saplings and adults, this could be a signal of a local extinction in the future. On the other hand, if we measure a population in a distribution limit and we measure high regeneration and high levels of growth and adult growth on growth in saplings and adults, this could be a signal of a potential expansion of the distribution. These demographic rates could give us information about stabilizing mechanisms. For example, if in a population we record very low growth for both saplings and adults, but if the regeneration is high, this could be a stabilizing mechanism that could allow the population to persist during time. In a similar way, if we record in a population low regeneration and low sapling growth, but if the adult growth is high and the adults lives for a long time, could also suppose another mechanism of persistence for the population, at least for some time. So mortality and longevity is something that can be also easily measured. It is also necessary to account for biotic interactions that can affect plant performance, such as herbivory and competition. And both altitudinal and latitudinal gradients can be sampled to analyze potential upward and northward migrations. Here there is a potential sampling indexing that could be performed with Pinus ponderosa, measuring these demographic rates along the latitudinal gradient. For example, at the leading age, at the northward distribution limit, could be in British Columbia, Washington, or Montana. Another place to sample could be at the core of the distribution in Nevada or California, and a rare age when a retraction can be expected could be here in Arizona. So there are many problems with this potential sampling indexing because differences in ponderosa varieties along its distribution range can make difficult to compare sample population. Also, differences in growing season and precipitation seasonality between sample population can also make difficult to compare them. And biotic factors such as herbivory and competition can change significantly across the latitudinal gradient. So I'm not sure if it's possible to perform this indexing. So if you have any idea you want to share, you're welcome. And this is all. Thanks for your attention, and sorry about my English.
All right, we have some time for some questions.
Yeah.
Going back to the first study with the saplings, can you tell us more about the regeneration process at the site? So were the saplings, were they planted or were they natural regeneration?
Yeah, this area was burned like, I don't know, maybe 20 years ago, and it was a reforestation experiment there in the area to test the potential use of nurse plant, nurse shrubs. So they are planted 12 years ago. So I think that they are like, yeah, I think 10 years old saplings. All of them are of the same age, and they live in the same environmental conditions. They are inside some fenced areas to avoid herbivories, ungulates, because browsing is a severe problem in this area. So I think that it is a good situation to test the response to drought.
So just one other question. Do you know about the genotype of the planted seedlings, the seed source? Are they local?
Yes, are local, are just from the area, just surrounding just 1 meters around it. They collect the cones and seeds, so are supposed to local adapted.
My question is kind of similar. I'm thinking about the third study where you're comparing junipers
Mhm.
What what part of the demography of the juniper makes it more resilient to drought or to fire than the plant species? Like why is it more resilient?
Okay, here we have data only about shoot growth and needle length. I only saw in the slide, I only saw the data of shoot growth of branches. We took like, for juniper is like 10 branches per individual, but we have also data about regeneration. And for Juniperus oxycederus at the low elevation, they have, I mean, good sapling and seedling density, but Juniperus communis at high elevation, the regeneration potential is quite low. So maybe it will have some problems in the future because they need like very humid locations for seedlings to establish. And the good thing with juniper is that they can, I mean, growth again when you cut or with fire, with fire. So are very resilient in this sense.
Is this species of juniper a sprouting species? It sprouts from the root system?
Yes.
Okay.
Yeah.
I got a question just about, you mentioned that your, included that Baylor's small control, control for plant scenario was associated with the slower mortality. And I guess I was just curious, what exactly is the model control used for? I know there's different strategies often that different trees take in place. What do you mean by small control?
I mean, you are asking me about because why stomatal control could be related with lower vulnerability to drought?
Yeah.
This is more or less. Well, okay. In the beginning, when we start to do this study, there are like Nate McDowell that works with some mortality mechanisms. State that there are like two mechanisms of drought mortality: one hydraulic failure, and the other one carbon starvation. So we think that maybe stronger stomatal control and stronger closure of stomata could be related with carbon starvation, but there are now new studies that say that both processes of hydraulic failure and stomatal and carbon starvation are very interdependent. So really, we cannot say nothing. I mean, we have two results. The mortality rate is lower for is lower for Pinus nigra, and Pinus nigra present greater stomatal control. So we think that there are some relation. Maybe it's able to use water more efficiently, conserve more water. So maybe it could be an advantage during droughts. And Pinus sylvestris, it seems that use water less, I mean, with less control, don't reduce the drought, the growth during droughts. So maybe it could be more vulnerable. But also it's like a hypothesis and a suggestion. But there are other studies with similar results that link stomatal control with mortality.
I think in your first study, you had some data on the oxygen 18 isotope in beetles. I know that locally, it's common to use that as an indicator of the seasonality of water use. I can't remember which way it's enriched, but either winter or summer is enriched in oxygen 18. Do your results have anything to do with the season of water use? Or how would I interpret, how do I interpret your oxygen?
Vamos a ver. Yeah, we know that maybe our results are influenced because of in 2005, temperatures were higher than in previous years, and maybe I mean there are more evaporation in water, so this could enrich this water and increase the oxygen 18. However, we think that the difference in between years are very high for oxygen and also for carbon 13. So we think that at least in part they are derived because of the stomatal conductance reductions. I mean, these data of carbon 13 are very high. If we compare with other pine species of more dry areas, they don't record these very strong, I mean, these very high values. So I agree with you that maybe the oxygen 18, maybe some part is because of evaporation. I mean, the water source is like more enriched in oxygen 18, but the source will be the same because we don't have really many precipitation in summer. So I don't know if I answered correctly. Or
I don't know either, but thanks for trying.
In your second study, you mentioned that gradient growth is more affected by temperature than precipitation, and you said that that might be attributed to some microclimate effects. Could you go more into depth on what type of microclimate effects would lead to that finding?
Okay. I mean, the best for explain this is the first photograph. I mean, these are relic populations of these pines that are surrounded by reforestations, and so I think that these populations survive maybe in the most in the most appropriate place to live. These pines, in summer in summer there is a very dry place with very high temperatures, but in winter, apart from precipitation, there are many fog. I mean, horizontal rain. You can see in the photograph. So maybe this could be. I mean, it's only an idea, but I mean, I work many days in winter and I cannot see nothing about 1 meter. So it would be like another source of precipitation that we don't really include in the study, and could have a role. I don't know.
Any more questions? All right. Do you have anything else you'd like to talk to us here about? We will be having a discussion in room 34 here shortly, and then after that, shortly after 6, we'll be at Lumberyard for beers and food. So if you have any interest in joining us for either one of those,